Very glad to meet you, students. Today let us learn about chapter 4, Principles of Inheritance and Variation in 12th standard Bio-zoology. This chapter we will discuss about genetics, multiple alleles, example ABO blood grouping, RH factor, sex determination in human, insects and birds, sex-linked inheritance, karyotyping, and finally Mendelian disorders. Examples: thalassemia, albinism, phenylketonuria, Huntington chorea. Genetic is a branch of biology which deals with the study of heredity and variation. Actually, gene is the unit of heredity. Heredity means inheritance of characters from one generation to another generation is called heredity. Actually, gene is inherited factors which determine biological expression of an organism, both morphologically as well as genetically. Variation is the differences among the individuals from parents to offspring. Important topic: multiple alleles. In Mendelian research, all the genes have two alternative forms: dominant, recessive. The dominant mostly represented as the capital form. The recessive mostly expressed in the form of homozygous condition. Example: tall versus dwarf. When three or more alleles of a gene control particular trait, the homologous chromosome is called as multiple alleles. This kind of inheritance is called multiple allelism. Example: ABO blood group in human beings. Blood group differs from person to person based on the presence of antigen and antibodies. Carl Landsteiner discovered two types of antigen, A antigen and B antigen, on the surface of RBCs. Blood group actually determined by presence of antigen and antibodies. The person is having A antigen, the person may be considered as A group. If there is no antigen in the blood group, it is considered as O blood group. There has only antibodies. A blood group person having A antigen, B antibodies. B blood group person having B antigen, A antibodies. In AB group, AB antigen will be there, no antibodies. In O group, there is no antigen, only AB antibodies. Actually, there are three autosomal alleles located in chromosome number 9, which determine the blood group of the gene controlling blood is called L, after the discovery of Landsteiner. Or I is used for the allelic forms. I means isoagglutination. There are three allelic forms: IA, IB, IO. ABO blood group are controlled by three allelic forms. They are IA, IB, and IO. So both the IA and IB allele produce transferase enzyme. They are N-acetyl galactose transferase. But IO do not produce any terminal enzyme, so they are called null allele. ABO blood groups, there are four phenotypic: A, B, AB, then O. Then possible genotype of ABO blood grouping. Next chapter, first it is discovered by Carl Landsteiner and Alexander Wiener in rhesus monkey. So they are called as RH factor. It is found in surface of RBC. RH factor refers to immunogenic D antigen. If a person is having RH antigen, the person is called as RH positive. If it is not having, the person is called as RH negative. Here there are two hypotheses regarding genetic factor, especially RH factor. The first one, Fisher and Ray's hypothesis. In this hypothesis, there are three pairs of alleles located in three different loci. In this hypothesis, capital D dominant allele will produce RH positive type. Double recessive small double d will produce negative phenotype. Then another hypothesis, Wiener hypothesis. In this hypothesis, there are 8 alleles located in single loci. So all dominant allele produce RH positive type. Then all all recessive types produce RH negative type. Here, capital represent dominant allele, small r represent recessive allele. So dominant allele always RH positive, then recessive allele or small r represent RH negative factor. Erythroblastosis fetalis or hemolytic disease of newborns, which resulting in blue jaundice and anemia. Actually, RH compatibility is very important in childbirth. RH negative mother and RH positive child, no problem in the first pregnancy. During second pregnancy, at the time of delivery, the problem will occur. The hemolytic disease will happen because at the time of first delivery, injury will happen. The RH antigen will enter into the mother's body. Next topic, sex determination. Sex chromosomes are called allosomes, which determines the sex of an individual. They may be homomorphic or heteromorphic. Actually, homomorphic produces only one type of gamete, so they are called homogametic. Heteromorphic produces two types of gametes, so they are called heterogametic. Example, XX, XY. Heterogametic males. Heterogametic males mostly produce dissimilar gametes. It is mostly seen in type of organism. Females are homogametic in nature. XX. Males XO or XY. AA represented as autosomes or body chromosome or somatic chromosomes. Allosomes, they are called as sex chromosomes. XX, XO type of sex determination. In this type of sex determination, female having 2X, male having only one X chromosome. During gamete formation, only one gamete produced, other it is not having. Other is not having chromosome. Next, XX, XY type of sex determination can be seen in Drosophila and human beings. In this type of sex determination, 2X chromosome represented in female. In male, 1X and 1Y chromosome will be there. Female. In heterogametic female, male possesses 2X chromosome. In female, they have dissimilar gamete. Is that W mostly used for instead of X and Y. The female produces two types of eggs. ZO, ZZ type, and ZW, ZZ type. ZO, ZZ type mostly is seen in moths, butterflies, chickens. In ZW, ZZ type, the female have alternative forms of alleles. ZW. The male have the same type. In WZ is the types mostly seen in gypsy moth, fishes, reptiles and birds. Actually here female have different types of gametes, so they are called as heterogametic female. Male organism having homogametic males. Human beings, there are 23 pairs of chromosomes. The first 22 pairs are autosomes. The remaining one pair is allosome. Female having similar type of chromosomes, so they are called homogametic females. Males have different types of alleles. Heterogametic males. In human, allosomes only determine the sex of an individual. For example, male parent produce two different types of gamete X and Y. Female parent produce similar type of gametes XX. Then the first generation itself, if it is individual having different types of gamete, it may be considered as male individual.